the broad classification of microorganisms. So, in this topic, we'll be learning about different categories of microorganisms and their characteristics. To begin with, first one is bacteria. In general, there are five different types of microorganisms: bacteria, algae, fungi, protozoa, and viruses. We'll be studying more about prions and viroids as well. So, we have learned about Whitaker's five kingdom classification system, in which bacteria are classified under Monera kingdom. So bacteria are unicellular prokaryotic microorganisms. And what are the characteristics of a prokaryotic organism? We have already learned in the diagram. Bacteria are classified as eubacteria. Eubacteria, it involves cyanobacteria, that is photosynthetic bacteria, actinomycetes, mycoplasma, rickettsia, chlamydia, and other types of microorganisms or bacteria that we'll see in next few slides. Another class of bacteria is archaebacteria. Archaebacteria are the bacteria which grow under extreme environmental conditions. That is the basic difference between eubacteria and archaebacteria. Eubacteria are generally existing in normal environmental conditions. Archaebacteria are existing in extreme environmental conditions. The first characteristic of bacteria is size. Bacteria exist in various sizes and various shapes. Their size is measured in terms of micrometers. So, in general, the spherical size bacterium, it measures in diameter ranging from 0.75 to 125 micrometers. A rod-shaped bacterium will have in general a diameter of zero point 0.5 to 2 micrometers, and other may have more than 100 micrometers in length. So these bacteria, they have found in various sizes and shapes. The spherical shape bacterium, the spherical shape is called as coccus, and in so coccus is singular and cocci is plural. They exist in rod shape, which is also called as cylindrical shape, or bacillus, which is in singular. Bacilli, plural. So if it is bacillus, it indicates the shape of the organism is rod shape. Bacteria may be of different shapes like square shaped bacteria, rounded bacteria, or pointed ends bacteria. For example, the exceptional shapes are there. For example, Stella. Stella is the name of a bacterium that has star-shaped cells. Pasteria has pear-shaped cells. Caryophanon has disc-shaped cells. Few bacterial species does not have a definite shape. They are called as pleomorphic organisms. The component of bacterial cell that decides the definite shape of bacterium is cell wall. So if cell wall is present, then that bacterium has a definite shape. If cell wall is absent, then there is no specific shape, sorry. These bacteria are, or that shape is called as pleomorphic. These are the various shapes of bacteria. Spherical shape, that is cocci. So bacteria, so this figure, it indicates or it shows the arrangement. That is the next characteristic. First one is size, second one is shape, and third one is arrangement. These bacteria may exist as a single cell, or it may exist as a pair of cells or a group of cells. That is called as arrangement. So that is dependent on the plane of their division. So in what plane these organisms are asexually reproducing, that plane decides their arrangement. So these arrangements are two two cocci arranged like this. It is called as diplococci, which is a characteristic of Streptococcus pneumoniae, and which is a characteristic of Neisseria meningitis. Another arrangement is these cocci are present in chains. This arrangement is called as streptococcal arrangement. Strepto means in chains. The example of bacterium is Streptococcus pyogenes. So from the name you can guess what is the arrangement, and that is why the name of this bacterium is given. If it exists in chains, then the bacterium's name, it starts with Streptococcus. Next arrangement is tetrad. That means four cocci are arranged in a group. If more than that arranged in a group, like a group of grapes, it is called as staphylococci. Staphylo means in groups, and therefore the name of the bacterium may be given as Staphylococcus aureus. So why Staphylococcus aureus? Because it exists in groups. Then Sarcini. The example is, so small groups are called as Sarcini. For example, Sarcina genus. In case of rods, there are arrangements like these rods are attached in a chain. It is called as streptobacilli. Strepto means chain, bacilli means rod. So this is the particular arrangement that can be seen in case of bacterium called as Bacillus anthracis. Flagellated rods. These are the rods which has flagella as a motile organ, as a organ for motility. That is a characteristic of Salmonella typhi. It is a bacterium that causes typhoid fever in humans. Spore forming Clostridium botulinum. So these bacteria are rod shaped bacteria. You can see the yellow part. That yellow part is the presence of endospore. Spore present inside the cell called as endospore, which is one of the organelles of bacteria that we'll be discussing later on. Other arrangements that can be seen, or other types of bacteria which are showing characteristic shapes, are spirals, vibrios. Spiral shaped bacteria, the shape is called as vibrio, and therefore the name of the bacterium is Vibrio cholerae. Vibrio cholerae is the causative agent of cholera disease in humans. It has a typical arrangement of a single flagellum at one end. It is called as polar flagellum. These bacteria are actively motile. Others are spirilla. Example is Helicobacter pylori. Helicobacter pylori has this kind of structure, spirillum type of structure, and it has flagella, more than one flagella at one end. These bacteria are extremely motile, and these bacteria are existing or they may be responsible for stomach ulcers. You remember in 2005 or so, the scientists they discovered this organism as a cause of stomach ulcer, and for this they received a Nobel Prize. Another category is spirochetes. These bacteria are also extremely motile, but you cannot see the exoflagellum in this case. But still, these bacteria are motile because they have flagella inside the cell. That means the flagella is present between cell wall and cell membrane, and these bacteria are extremely motile, and these are human pathogens also. For example, Treponema pallidum is a human pathogen responsible for syphilis disease. This is a very interesting, fascinating example of star-shaped bacterium. You can guess its size. It is approximately 3 micrometers long. It is star-shaped bacterium, that is stela. This is the another figure of that organism, and this is one more variety of star-shaped bacterium which has tentacles like this. All these are microscopic images. So you can see that this star-shaped bacterium has a size of approximately 30 micrometers due to presence of these tentacles. Another fascinating example is these are the pleomorphic bacteria. This is a single culture. This is a single type of bacterium, but still its cells are of different shapes and sizes. So these different shapes and sizes is because there is absence of cell wall. So absence of cell wall, that's why these cells are pleomorphic, which is a characteristic of organisms like mycoplasma. Next characteristic is reproduction. Reproduction means it can be divided into two categories: asexual reproduction and sexual reproduction. There are three ways of asexual reproduction in case of bacteria. First one is binary fission. So this is the method by which bacteria, most of the bacteria in general, they reproduce to produce two cells from one cell. The first step is whatever the genetic material and whatever the biochemical material, biochemical machinery which is present inside the cell, it is doubled. So the first step is elongation of cell, and the genetic material or the chromosome, bacterial chromosome, it gets duplicated. In the second step, the cell wall and plasma membrane, it begins to divide. So there is formation of a septa, there is formation of a compartment that grows inside cell wall and plasma membrane. It grows inside, and finally it leads to formation of a cross wall. And Here we can see that there is complete division of cytoplasm, there is complete division of nuclear material, and finally these two cells they separate. This is how is the mechanism of binary fission. Bi means two. Another way of asexual reproduction that is not general, it is fragmentation. So fragmentation is seen in case of photosynthetic bacteria, or fragmentation is seen in case of that particular class. So in this case you can see that fragmentation involves separation of a small fragment is detached from the bacterial cell, and then that fragment it can grow independently to form a whole cell. Fragmentation is a common mechanism that is seen in case of fungi and algae, but some bacteria they also show fragmentation type of asexual reproduction. So this is particularly seen in case of filamentous bacteria. The third way of division, asexual reproduction, is budding. This is also not common in bacteria. There is a class of bacteria called as budding bacteria. So their unique characteristic is the method of asexual reproduction is budding. So in this case, this is generally seen in case of yeast, but in some bacteria, this mechanism is also seen. So this first step is the nuclear material is duplicated, then that nuclear material it move towards one end, and that end is a growing end which is called as bud. This bud then enlarges as the biosynthesis progresses. Cell wall synthesis occurs, cell membrane synthesis occurs, other material that is present in cytoplasm it is duplicated and it move towards the young bud, and finally after the full growth this bud it detaches from its mother cell. It is called as daughter cell. This is how the mechanism is existing in few bacteria. Another method of reproduction is sexual reproduction, which occurs rarely. The method of sexual reproduction in bacteria it is called as conjugation. This conjugation it requires cell to cell contact. Here there is an example of what happens in conjugation is genetic material from one bacterium is transferred to another bacterium. Genes are transferred from one bacterium to another bacterial cell. It is called as conjugation. So in conjugation, towards left hand side, you can see the cell is having a, a chromosomal DNA that is shown in green blue color, and it also shows the presence of a plasmid which is called as F plasmid. F stands for fertility factor. So this organism has this F plasmid which is shown in pink color. So this is the extra chromosomal DNA that is present in the cell. So this cell will be called as F plus cell or the donor cell. So this donor cell comes in contact with recipient cell towards right hand side. It is a recipient cell which is called as F minus. Why it is called as F minus? It can be seen in the figure. There is absence of F plasmid. So these two cells come in contact with each other through a specific attachment that is called as pilus. This pilus is called as sex pilus. So with the help of this sex pilus, these two cells come in contact with each other, and this is how it leads to formation of a bridge. In this formation of bridge, the cell wall and cell membrane it dissolves, and there is formation of a channel. You can see that this F plasmid, that is pink colored pink colored DNA, it is, there are two strands, so it is double stranded. So one of the strands it cuts, and that strand is received by F minus cell, which is also called as the recipient cell. So one of the strands from F plasmid it gets transferred from F plus cell to F minus cell. So this is how it is transferred. Transfer of one strand of F plasmid, and after the transfer is complete, again that strand it forms a new strand, complementary strand, so that again there is formation of a double stranded structure of plasmid. So finally you can see that the donor cell regains its plasmid, and the recipient cell receives a new plasmid. This is how the recipient cell becomes F plus cells. Now both the cells are F plus. This is the method of conjugation in which the genetic material is transferred from one bacterial cell to another bacterial cell. Other characteristic is motility. Motility is ability to move. Bacteria can move or swim in liquids or semi-solid medium using flagella. There are some bacteria called as spirochetes which contains endoflagella. So we'll be discussing more about this in the last chapter. There are some bacteria that can move in absence of flagella. The motility is called as gliding motility. Now, the class of eubacteria. Eubacteria are the class of bacteria uh, that grows under natural environmental conditions, general environmental conditions. These general environmental conditions are currently whatever the environmental conditions we are experiencing. So normal environmental conditions like 25 to 30 degrees Celsius temperature, water bodies, soil, rhizosphere, human body, etc. So the bacteria which are existing in this particular environment are eubacteria. So they are generally uh, existing under natural environmental normal environmental condition. I'm sorry, it is normal environmental condition. Eubacteria are divided into three categories: gram positive, gram negative, and mycoplasma. The basis of their classification is presence or absence of cell wall. Mycoplasma is the group in which there is absence of cell wall. If bacteria has cell wall, then there are two groups, gram positive and gram negative. What is the difference between gram positive and gram negative? Gram positive, so the difference lies in the cell wall, composition of cell wall, type of cell wall, content of cell wall. Cell wall of a gram positive bacteria has high percentage of peptidoglycan and low percentage of lipids. In contrast to that, gram negative bacteria have high percentage of liquids and low percentage of peptidoglycan. You need to remember this. So examples of eubacteria are the bacteria which are important in industries, which are important in fermentation technology, industrially important bacteria. that produce alcohols, antibiotics, organic acids, enzymes, fermented food products like curd, yogurt, cheese, etc. Examples of eubacteria are the bacteria which are essential in digestion of waste material or recycling of waste. The examples of eubacteria are the bacteria which are responsible for animal infections or human infections. For example, common infections are streptococcal sore throat, tetanus, plague, cholera, typhoid, dysentery, tuberculosis, leprosy, etc.